In the spring of 2013, construction workers in the German city of Erfurt were converting an old granary into a multi-story parking garage when their machinery struck something that stopped the entire project cold. Bones, human bones, dozens of them, packed into the earth beneath the foundation in a pattern that was unmistakably deliberate. Within hours, archaeologists were on site, and within days, the discovery had a name. This was the medieval Jewish cemetery of Erfurt, a burial ground sealed beneath the city for nearly 600 years, ever since the Jews of Erfurt were expelled in 1454, and a granary was built directly on top of their dead to erase the ground where they had been laid to rest. Now the past was breaking back through the concrete, and it created an immediate and delicate crisis. Jewish law is unambiguous about the dead. Graves are not to be disturbed. Bodies are not to be exhumed for study. For the archaeologists standing over that excavation site, the discovery was not simply a scientific opportunity. It was an ethical minefield, one wrong step from becoming a desecration. The Erfurt Jewish community itself had a long and turbulent history behind that buried cemetery. It was first established around the 11th century, grew into one of the largest and most prosperous Jewish communities in medieval Germany, and was then nearly destroyed in 1349 when a wave of anti-Semitic violence swept across Europe during the chaos of the Black Death, and most of Erfurt's Jews were murdered or driven out. Remarkably, a second community reformed just 5 years later in 1354 and rebuilt itself into one of the largest Jewish populations in the region, only to be expelled again a century later in 1454, after which the city built directly over the site of their cemetery as if to erase the memory of it entirely. Rabbinical authorities were consulted immediately after the 2013 discovery, and after careful deliberation, they granted a narrow and specific permission. Scientists could examine bones and teeth that had already become detached from the skeletons during 13th and 14th century burial practices and the passage of 6 centuries underground, but nothing more, and every individual studied would ultimately be reinterred with full dignity in a 19th century Jewish cemetery nearby. It was under that fragile agreement that a team of more than 30 scientists, led by Shai Carmi of the Hebrew University of Jerusalem and David Reich of Harvard Medical School, extracted genetic material from the teeth of 33 individuals who had lived and died in Erfurt in the 1300s. What they found, when they sequenced that ancient DNA, turned into one of the most significant discoveries in the entire history of Jewish genetics. And it was only the latest chapter in a much larger and more staggering scientific story, one that had already reached back thousands of years earlier to answer a question that no religious text, no ancient chronicle, and no archaeological dig alone had ever been able to fully settle. Where did the Jewish people actually come from, not as a matter of scripture or legend, but as a matter of biology, written into the actual molecules inside human bone? For thousands of years, a civilization lived in the narrow strip of land wedged between the Mediterranean Sea and the Jordan River. They built walled cities on hilltops, traded copper and cedar across the ancient world, developed writing systems that would eventually shape every alphabet used in the West, and buried their dead with offerings in the dark beneath the earth. Across the centuries, they were called different things: Hebrews, Israelites, Judeans, and eventually the world came to know them as the Jewish people. And for millennia, wherever history scattered them, they carried one persistent question. History gave partial answers. The Hebrew Bible offered its own account, beginning with Abraham called out of Mesopotamia toward a promised land. Archaeology gave fragments, broken pottery, ruined walls, inscriptions on stone. Linguistics traced the spread of Semitic languages across the ancient Near East. But none of these tools could look directly into the biology of a people and trace their ancestry backward with real scientific precision. That capability only arrived with modern genetics, and specifically with the ability to extract and read ancient DNA, the genetic material preserved inside bones and teeth of people who died thousands of years ago. When that tool was finally turned on the question of Jewish origins, the results were more illuminating, and in places more surprising than almost anyone expected. In laboratories from Harvard Medical School in Boston to Tel Aviv University in Israel, from the Max Planck Institute for Evolutionary Anthropology in Leipzig to the Hebrew University of Jerusalem, geneticists spent years running a very specific and tightly controlled test. Take the DNA of modern Jewish populations from every major community across the diaspora. Compare it against the genetic fingerprints of every ancient civilization whose remains have been recovered from the archaeological record. Which ancient people, if any, would turn out to be the closest genetic match? Researchers tested ancient Egyptians, who left behind some of the richest archives in the ancient world. They tested Mesopotamian populations from between the Tigris and Euphrates. They tested Anatolian farmers who first carried agriculture into Europe. They tested Bronze Age steppe pastoralists from the Russian grasslands, whose migrations, roughly 6,000 years ago, reshaped the genetics of half of Eurasia. They tested ancient Persians, Greeks, Romans, North Africans, Arabians, and peoples from the Caucasus Mountains. Across every one of those comparisons, one civilization kept emerging as the strongest and most consistent match. Not a marginal statistical suggestion, but a repeated, undeniable genetic connection. Not the Egyptians, not the Mesopotamians, not the Indo-European migrants who swept across the ancient world in successive waves. The ancient population, whose DNA most closely matches the genetic core of Jewish communities from every corner of the diaspora, is the Bronze Age population of Canaan, the very civilization whose world forms the backdrop of the earliest biblical narratives. This is not a fringe finding tucked into a footnote. The central study behind it, published in the journal Cell in 2020 by a team including Lily Agranat Tamir, Shai Karmi, Israel Finkelstein, and David Reich, sequenced genome-wide data from 73 ancient individuals unearthed at 5 archaeological sites across the southern Levant, later combined with earlier published genomes to build a dataset spanning 9 sites altogether in what is now Israel, Jordan, and Lebanon. These people lived roughly between 2400 and 900 years before the Common Era, deep in the Bronze Age and into the early Iron Age, precisely the window during which scholars believe ancient Israelite identity was taking shape. Their remains came from sites whose names read like a roll call of the biblical world. Megiddo, the fortified city that controlled the most important trade corridor in the ancient Near East, and whose name gave rise to the word Armageddon. Hazor, the largest city in the entire region during the late Bronze Age, described in the Book of Joshua as the head of all those kingdoms. Ashkelon, the coastal city whose excavations have produced some of the most extraordinary archaeological finds of the last century. Ain Ghazal in Jordan, one of the oldest and largest Neolithic settlements ever discovered anywhere on Earth. When researchers extracted genetic material from bone and tooth samples at these sites, purified it, sequenced it, and ran it through sophisticated statistical models built to measure genetic distance and shared ancestry, the results were strikingly consistent. Most present-day Jewish populations, despite their extraordinary geographic and cultural diversity, trace at least half of their ancestry back to those Bronze Age Canaanite populations of the southern Levant. Half is not a marginal or ambiguous signal. It means that if you could take a Jewish person from New York or Tel Aviv or Tehran or Addis Ababa and place them beside one of those ancient individuals excavated from a Bronze Age grave at Megiddo or Hazor, the genetic relationship between them would be stronger than the relationship between that same Jewish person and any ancient Egyptian, Mesopotamian, Greek, Roman, Persian, or Anatolian population ever tested. Of every ancient civilization run through this comparison, only the Canaanites produced that level of match. David Reich, one of the world's foremost experts in ancient DNA, put it plainly when he said the data points to a deep genetic connection between many Jewish groups today, across the entire diaspora in this part of the world, stretching back thousands of years. To understand why that finding is so remarkable, it helps to understand what the Canaanites actually were. In the Hebrew Bible, the word Canaanite is used almost entirely to describe the population that occupied the land before the Israelites, the people the Israelites were repeatedly commanded to displace. That framing has shaped how the Canaanites have been remembered for centuries, as a doomed people who simply lost and vanished. But archaeology and genetics together have overturned that picture. The Canaanites were a sophisticated cosmopolitan civilization sitting at the center of the Bronze Age world, organized as a network of city-states, each with its own king, but sharing a broadly common material culture, a recognizable art and architectural style, trade routes stretching from Egypt to Cyprus to Mesopotamia, and a shared genetic ancestry. They were not genetically uniform in any rigid sense. The ancient DNA reveals subtle regional differences, with coastal sites carrying slightly different genetic profiles than inland settlements, but researchers describe them as forming a coherent ancestral cluster, a population whose members shared more genetic history with each other than with distant populations. And that population itself was the product of earlier mixture. The Canaanites descended from Neolithic farmers who had lived in the Levant for thousands of years, blended over time with migrants arriving from the direction of the Caucasus Mountains and the Zagros Range in what is now Iran and northern Iraq. This eastern genetic component increased gradually through the Middle and Late Bronze Age, and the data even shows this happening in real time at Megiddo, where a small number of individuals carry notably higher levels of Caucasus-related ancestry than their neighbors. Evidence of newcomers who settled among the existing population, even as their descendants grew up eating the same local foods. Understanding exactly how this analysis works matters, because the methodology is what gives it scientific authority. Geneticists compare populations by measuring allele frequencies, the rates at which specific genetic variants appear across different groups. Populations that are closely related share more of these variants at similar frequencies than populations only distantly connected. By comparing hundreds of thousands of such markers across ancient and modern genomes simultaneously, researchers can reconstruct how populations relate to each other across both time and geography. The specific technique used most often in these Jewish ancestry studies is called QPADM modeling, a form of quantitative admixture analysis that tests whether a modern population's genome can be statistically explained as a mixture of two or more ancient source populations, and in what proportions. When this model is run comparing modern Jewish populations against a wide range of ancient reference genomes, Bronze Age Levantine populations consistently come out as the single largest identifiable source component. No other ancient group, alone or in any combination, produces a better statistical fit. The Canaanites are not simply one plausible candidate among several. They are the best supported answer the data produces, tested repeatedly against every available alternative. The growing accumulation of ancient DNA from the southern Levant has also allowed researchers to trace genetic continuity across the transition from the Bronze Age into the Iron Age, exactly the period during which ancient Israelite identity is believed to have emerged from within the existing Canaanite world. What the evidence shows for that transition is not a dramatic population replacement. It does not show a genetically distinct outside group arriving and displacing the existing inhabitants. Instead, it shows substantial continuity, the same broadly Canaanite genetic pattern persisting into the Iron Age populations of the region. This lines up with what many archaeologists have argued for decades based purely on material culture, that the Israelites were not a foreign people invading from elsewhere, but an identity that emerged from within the existing population of the land through cultural and religious differentiation. In a very real biological sense, the Hebrews were, to a significant extent, the Canaanites, who began calling themselves something different. What makes this connection so compelling is that it holds across Jewish communities separated for more than 2,000 years, embedded in wildly different host societies and shaped by very different degrees of local genetic mixing. Take Yemenite Jews, who maintained a presence on the Arabian Peninsula for so long that oral tradition in the community claims ancestry reaching back to the era of King Solomon. Living in relative isolation from other Jewish communities for centuries, their genetic pool absorbed real influence from the surrounding population of the Arabian Peninsula. And yet, when researchers compare Yemenite Jewish DNA to ancient Canaanite genomes, the ancient Levantine signal is still unmistakably present underneath. Consider Mizrahi Jews, the communities of Iraq, Iran, Syria, and the wider ancient Near East, who lived in or near the Canaanite homeland continuously for over 2,000 years. Their genetic profiles consistently show higher levels of ancient Levantine ancestry than their Ashkenazi or Sephardic counterparts, a simple reflection of the fact that they never left the region and absorbed comparatively less outside genetic influence over the centuries. Study after study, run independently by different research teams, keeps landing on the same conclusion. Beyond the broader genome inherited from both parents, the story is also written along two much narrower genetic threads. The Y chromosome, passed almost unchanged from father to son, and mitochondrial DNA, passed from mother to child down the maternal line. The Y chromosome data is especially striking. Certain paternal lineages appear at elevated frequency across virtually every major Jewish community worldwide. One lineage, sometimes labeled J1, is strongly associated with Semitic-speaking populations of the ancient Near East and appears at particularly high rates among Cohen families, the Jewish men who trace a priestly lineage back to Aaron, the brother of Moses. A second lineage, labeled J2, is common throughout the Mediterranean and the Fertile Crescent and reflects shared ancestry with populations of the northern Levant. What is remarkable is how consistently these lineages appear across geographically distant Jewish communities. Ashkenazi, Mizrahi, Yemenite, and Sephardic Jews all show elevated frequencies of these same paternal markers, frequencies clearly higher than in the surrounding non-Jewish populations of Europe, North Africa, or Arabia, where these communities have lived for centuries. This pattern is difficult to explain through local intermarriage alone. If Ashkenazi Jews had simply absorbed the paternal lineages of the European populations around them, their Y chromosome profile would look far more European. Instead, it retains a distinctly Middle Eastern signature, tracing back toward the men who founded those communities when they first migrated into Europe out of the ancient Levant. The Y chromosome, in a sense, is the part of the genome most resistant to the diluting effects of diaspora, and what it preserves across all that diversity is an ancient Middle Eastern signal pointing back toward the same origin the wider genome identifies from a completely different angle. The maternal line tells its own surprising part of the story. A landmark 2006 study led by Doron Behar found that roughly 40% of all Ashkenazi Jews alive today descend from just 4 women, founding mothers whose mitochondrial lineages expanded dramatically within the community over the last 2,000 years. Unlike the Y chromosome data, Some of these specific maternal lineages appear to have prehistoric European roots rather than Near Eastern ones, suggesting that at some point early Jewish male migrants settled in Europe and had children with local women, whose descendants then multiplied inside a community that remained religiously and socially closed to outsiders for generations afterward. It is a small but telling detail, a reminder that the overall Levantine signal running through Jewish DNA is not a story of total isolation, but of a population that absorbed limited outside ancestry at specific points, and then locked that ancestry inside a tightly bounded endogamous community for the better part of 2 millennia. The Ashkenazi story is the most genetically intricate chapter of all. These are the Jews of Central and Eastern Europe, of Germany, Poland, Russia, and Hungary, the population from which so many of the last 2 centuries' most celebrated scientists, writers, and thinkers emerged. Their name comes from the medieval Hebrew term for Germany, and their identity as a recognizable community dates to roughly the 10th and 11th centuries, when Jewish settlers established themselves in the Rhineland cities of Mainz, Worms, and Speyer, later spreading eastward into Poland, Lithuania, Ukraine, and Russia, reaching an estimated population of 8 to 9 million people by the eve of the 20th century. This is where the Erfurt discovery becomes so important. The medieval Jews of Erfurt, alive 6 centuries before our own time, turned out to be genetically more diverse than modern Ashkenazi Jews are today. The 2022 study found evidence of at least 2 genetically distinct subgroups coexisting inside that single medieval community, one leaning more toward Middle Eastern ancestry, and the other more toward European ancestry, likely reflecting more recent arrivals from the East. That internal diversity has since essentially vanished. Modern Ashkenazi Jews, no matter where in the world they live, are strikingly genetically similar to one another, almost like an extended family, a uniformity explained by what population geneticists call a founder event. At some point before the 14th century, the Ashkenazi population passed through a severe genetic bottleneck, in which a relatively small number of individuals became the biological ancestors of nearly all modern Ashkenazi Jews. Genetic variants carried by that founding group, simply by chance, became elevated across the entire descendant population, including variants linked to serious disease. At least 8 of the 33 medieval Erfurt individuals already carried pathogenic mutations that today cause elevated rates of Tay-Sachs disease, Gaucher disease, and hereditary breast and ovarian cancer among Ashkenazi populations, a direct genetic echo of that ancestral bottleneck. But even within the Ashkenazi genome, layered with centuries of European admixture and geographic distance from the Middle East, researchers still find that somewhere between roughly 1/3 and 2/3 of the ancestral makeup traces back to Near Eastern origins. The European component is real and substantial, but it sits on top of a Middle Eastern foundation, a compressed, partly mixed, but still statistically readable signal tracing back every time to the ancient Levant. The science of ancient DNA also forces a reckoning with how incomplete earlier reconstructions of this history really were. For generations, the origins of the Jewish people were debated almost entirely through texts and artifacts, biblical narratives, Egyptian inscriptions, Assyrian and Babylonian records, archaeological layers of burned cities and rebuilt temples. These sources are invaluable, but they are fragmentary and filtered through the particular agendas of whoever created them. Egyptian scribes recorded victories and rarely defeats. Biblical authors wrote about covenant and faith, not population genetics. Assyrian kings inscribed their conquests, but had no interest in the ancestry of the people they conquered. Genetics does not replace any of that evidence, but it adds a dimension immune to those same filters. A tooth does not carry a theological or political agenda. The genetic signal encoded in ancient bone is simply the accumulated record of who had children with whom across thousands of years, deposited in the chemistry of ancient cells and waiting to be read. It is also worth being clear that Jewish populations are not the only inheritors of that ancient Canaanite ancestry. A separate 2017 study led by Mark Haber found that modern Lebanese populations can trace more than 90% of their ancestry to Bronze Age Canaanites. Other research indicates modern Palestinian populations carry a similarly high proportion of ancient Levantine ancestry, among the highest levels of genetic continuity documented anywhere in the ancient DNA literature. The Canaanite genetic signal runs through the genomes of multiple modern peoples, Jewish, Arab, and others, each of whom followed a radically different historical path while carrying the same underlying biological inheritance. This shared ancestry has, unsurprisingly, become entangled in modern political rhetoric on more than one occasion, with different leaders in the region invoking the Canaanites as symbolic ancestors to support competing claims to the land. The genetic data itself does not adjudicate those political or moral arguments. It simply describes what happened biologically across thousands of years in one of the most contested pieces of land on Earth. And what it shows is not a clean story of one group replacing another, but of ancient populations transforming and persisting. their biological inheritance flowing forward into peoples who today think of themselves as entirely distinct from one another. Perhaps the most striking implication of all this research is what it reveals about the nature of identity itself. The Jewish people have maintained one of the most coherent and continuous cultural identities in recorded history, built around shared texts, shared law, shared memory, and shared practice, even while undergoing some of the most extensive genetic mixing of any population on Earth. Levantine ancestry moved through Europe and absorbed European ancestry along the way. It moved through North Africa and absorbed North African ancestry in return. It did the same across Persia, Yemen, Ethiopia, the Ottoman world, and the Russian Pale of Settlement. And through all of that movement and mixing, the community remained recognizably itself, not because its genes stayed fixed, they did not, but because its culture, its language of prayer, its calendar, and its collective memory were maintained with extraordinary discipline across every generation. The genetics does not diminish that achievement. If anything, it makes it more remarkable. A coherent identity sustained across so many centuries of genetic transformation, so many host societies, and so many pressures toward assimilation or disappearance. There is something almost difficult to fully absorb in what these findings represent. Actual biological molecules have persisted through 4,000 years of upheaval, through the collapse of the Bronze Age world around 1200 BCE, when the great civilizations of the Eastern Mediterranean fell within a single generation, through the Assyrian conquest of the northern kingdom of Israel in 722 BCE, the Babylonian destruction of Jerusalem in 586 BCE, the Persian restoration, the Greek conquest under Alexander, the Roman destruction of the Second Temple in 70 CE, the dispersion across the known world that followed, the medieval centuries of persecution and expulsion, and the Holocaust, which destroyed roughly a third of the world's Jewish population in less than a decade. Through all of it, across all 4,000 years, the thread of ancient Canaanite ancestry persisted in the Jewish genome, stretched and layered with the genetic inheritance of a dozen other civilizations encountered along the way, but never entirely severed, waiting inside bone and tooth to be read by a laboratory that would not exist for another 4 millennia. None of this science can explain what it means, in lived experience, to be Jewish. It cannot capture the weight of a Sabbath candle, the sound of a shofar, or the meaning of a Torah scroll read aloud by people who know they descend from those who read the very same words 100 generations before them. That belongs entirely to the tradition itself, sustained through circumstances that erased nearly every other ancient civilization that faced comparable pressure. But science can now say something that theology, archaeology, and linguistics alone could never fully establish on their own. The Jewish people carry within their biology a genuine, measurable, scientifically documented connection to the land and the civilization that gave rise to the world described in their oldest texts. Back in that reclaimed cemetery in Erfurt, in the quiet return of 33 sets of remains to consecrated ground after their teeth had given up one final secret, the discovery reached back across 6 centuries. And through the deeper science that made sense of it, it reached back across 4,000 years more, all the way to the walled cities and trade roads of Bronze Age Canaan. When a Jewish person anywhere in the world today reads about the ancient cities of Canaan, the fortified hilltops, the busy trade routes, the diverse peoples of that narrow strip of land between the river and the sea, they are reading, in a small but real and measurable sense, about their own ancestors. 4,000 years of exile and return, of dispersion and memory, of an identity carried across every ocean and through every catastrophe the world could produce. And at the end of all of it, the DNA still says the same thing. The thread holds.